All right, so we're gonna take a little look at the microscopic world, which is, I have a fairly substantial background in microbiology and cell biology, but this is one of the areas that just always really appealed to me, and I think it's one of the areas that probably has the most of the examples from intelligent design can be found right within the microscopic world. And that's interesting because no one's ever seen it before. We're the first generation to ever see the microscopic world. The first generation that ever had technologies that would allow us to view things of a microscopic scale. We're going to look at things today that no one has ever seen before. Evidences of design, evidence of God's creation, things that He made that no one's ever seen before. Much like the vast reaches of the cosmos, though, as well. You know, up until our generation, no one has ever seen the cosmos except for the stars that were near us. They were able to see the stars in our own galaxy, but nothing beyond that. Now we know that there are at least 100,000 galaxies with 100,000 stars each out there in the cosmos. No one has ever seen that part of the cosmos. No one's ever seen the microscopic world either. I am kind of fond of saying that it seems like God created the cosmos in such a way that we would never finish fully investigating it. The better our technology gets, the more of His creation is revealed to us, and the more amazing, therefore, it seems. Now we know the cosmos is much bigger than anyone thought it was before. And we know that there's this microscopic realm. And as we get down to this microscopic realm, we see complexity that no one could have dreamed of before now. A level of complexity and sophistication of design no one could have dreamed of before now. I do have a fairly substantial background in cell biology. I've worked in a number of research labs over the years. I don't know if you can recognize me in the picture here because since that picture was taken, I kind of turned into Shrek a little bit, you know what I mean? Kind of took on a Shrekish, physique and all. But that picture was taken when I was working at a lab at Texas Tech University. The cotton fiber production lab at Texas Tech University was tasked with trying to grow cotton cells to lengths that would allow them to be used by textile industries. So instead of growing cotton fiber on farms by growing plants, we would grow tissue. We're using tissue culture, isolating cotton cells and growing these in large fermenters, large bioreactors, where we would provide the nutrients and the hormones to try to trigger them to grow to lengths that would allow them to be used for textiles and all sorts of things. But and as part of this job, amongst others, I've spent a lot of time on the microscope. A lot of time on the microscope. I don't know if I spent a year of my life on a microscope, but it kind of seemed like that at some point. Checking our cultures for contaminations. I ran the picture was showing the dean of the arts and sciences there, Joe Gooden, with it was a more publicity photo with the new image analysis system that we bought, where I would use this system to measure the lengths of cells that we were growing in our bioreactors. And to my knowledge, they spent $50,000 on that little system that you see there, the microscope and the computer that's hooked up to it. And to my knowledge, I'm the only person that ever used it. It's true. But the microscopic world is a very beautiful thing. It's just a tremendously beautiful thing. There is an elegance to the way things move in the microscopic world, a symmetry and a beauty to it that's unlike anything else common to our experience. I kind of liken it to maybe, I don't know, it's not similar, but like scuba diving. Scuba diving is so foreign to our senses. When you get down in that water, you encounter a new realm, a realm so foreign to your normal experience that the microscopic world is similar in that regard. It's just so foreign to our normal senses. There's a tremendous beauty to it. And these single-cell organisms, like those that you're seeing here, these microorganisms are everywhere. The culture that you're seeing in this little video right here can be obtained almost anywhere. Any little puddle out there has single-cell organisms like this. If there's some organic debris, some dirt, and other stuff decomposing in a body of water, you can go down into that dirt and with a little eyedropper, suck up some of that, put it on a slide, and you will see some of these guaranteed. So think about that next time you're walking around your barefoot through mud and stuff, because you don't know. I mean, there's a lot of stuff in that mud, I'm just going to tell you. One of the ways that they create cultures like this, literally, if you want to create a culture that's great for microscopy with lots of these single-cell organisms, you take a handful of grass, Hay seems to work better. Take a handful of hay, put it in a container, fill it full of water, put it in a warm place, and leave it for like 2 weeks. Then you come back in, suck up some of that water, put it on a slide, and it's just teeming with stuff. The microscopic world is out there, and it's everywhere that you look in the world. And there is a beauty to it, an elegance. We'll look more at that as we go through this. But we also know that the microscopic world is the host of many of our diseases of old as well. Things like bacteria. This is the bacteria that causes food poisoning, Salmonella. Bacteria they're beneficial organisms. They're part of the decomposers. Bacteria and fungi are the organisms that break things down so that the materials that are being used by the present generation are back in the topsoil and available to the next generation. They're the decomposers. And they serve many beneficial roles in ecosystems in addition to that. Your stomach your large intestine, or what you call the colon, is completely full of bacteria that help you digest your food. They help the herbivores digest their food, either by way of the cecum or the rumen. But they also, because they're decomposers, if they get in a cut somewhere, they can start trying to decompose you before you're actually dead, so they can cause trouble. But many of the diseases of old, leprosy, we now know is due to a bacteria. Leprosy from the Bible, or ulcers are now known to be caused by a bacteria. Historically, though, when I was young, it was believed that ulcers were caused by stress. It was taught that if you stressed yourself out, if you had too stressful of a lifestyle, too stressful of a job, you could give yourself ulcers. And people that had an ulcer would have it for life and would have to drink or have antacids. People would walk around with bottles of antacids or little bottles of antacids that they were constantly having to consume because, an ulcer is a sore on the inside of your stomach. And because you have glands in your stomach that are secreting acid, you have hydrochloric acid in your stomach. And so a little sore in the stomach, exposed to hydrochloric acid is going to burn. So they would take antacids to neutralize the hydrochloric acid and not burn. And when I was young, I can tell you the names still. There was Maalox and Tums and Rolaids and Milk of Magnesia and all of these antacid brand names that I know because commercials were prevalent. But how long has it been since you've seen a commercial for an antacid? Once they discovered that the ulcers were caused by a bacteria, now all they do is give you a course of antibiotics, small, whatever it is, 2-week course of antibiotics, kills that bacterial culture that's in your stomach, the ulcer heals up, no more need for antacids, and it decimated an entire industry of antacid producers. But, bacteria are everywhere. They're tremendously beneficial roles that they play in ecosystem, but they do grow and divide very rapidly. This bacteria again is Salmonella, which under optimum condition, bacteria can grow and divide about once every 20 minutes. A bacteria can replicate itself about once every 20 minutes under optimum conditions. So they grow at such a rapid rate, it's hard for your immunity system to keep up with that. And that, like I say, you get a bacteria in a cut, and one of the roles is to be a decomposer, and they will start trying to decompose you before you're actually fully dead. But one of my main goals here in talking tonight is to point to evidence of design within the microscopic world. Because today the scientific community says there is no evidence of design. They use the word design a lot. You hear biologists in their published papers talk about design that they observe in the biological world, but at the same time say it's only the appearance of design. It's not really design. It just has the appearance of design. It's actually evolved. So the question is, how can we tell? Can we tell if something is designed, truly? Can we tell through our senses, through experimentation? Can we tell if something is designed? And I argue that we can. Just crude observations alone are significant enough to tell us if something is designed. For example, on this graphic, if you're walking along a beach and you've come across something on a beach that is shaped in a peculiar way, could you tell whether that shaping was done by hands, by an intelligent mind who had shaped it into something like an arrowhead, rather than it being eroded into that shape by purely natural processes? Or you come across a sculpture like this one shown here. Come across a sculpture. Could you tell that sculpture had been made by hands? Every once in a while you can see a crude shape of a face, in some rocky outcrop or something, but you can tell the difference between something that was formed by an intelligent mind versus something that was formed through purely natural processes. This is the argument. Can we tell that things within the cell, that cells themselves, are the result of an intelligent mind that formed them, or are they the result of purely natural processes? I argue that just looking at cells alone is a powerful argument. Some of them are just so beautiful, and there's a symmetry to it. These are diatoms on this picture. Diatoms are a single-cell algae that have enclosed themselves within a cell wall made of silica. And there's just a tremendous beauty to these. I'll zoom in a little bit more so you can get a look at some of these. But you can see all the shapes of these diatoms. There's a tremendous beauty that we see in the microscopic world. I'd argue if you saw those anywhere else, at any other size, if you came across one of those, stumbled across one of those in a hallway somewhere or laying on the ground outside, that you would assume someone had made that some glass blower had made that, rather than that having been formed through purely natural processes. And if we look at these diatoms at even higher power magnification, it even speaks more to this. One of the most powerful microscopes that we have is the electron microscope. And there's a couple different kinds of electron microscopes. The one that makes these images, called the scanning electron microscope, gives us a three-dimensional look at these. And with these, you can magnify things up into the tens of thousands of power magnification. I'll give you a little bit better look at these. Again, these are diatoms. This is a single cell organism that you're seeing there of a tremendously small size. The scale that you show there shows 20 micrometers. That little U is a symbol for the micron, which means 1 millionth. So that's 1 millionth. That's 20 million. So just to give you a little better, you know how big a centimeter is? There's 10 millimeters in a centimeter. You can see a millimeter with the human eye, but just barely. You can't see much below a millimeter. It's about the width of a dime. I don't think you'd have struggled to see half a millimeter, but there's 1,000 microns in a millimeter. And what you're looking at there is just 20 microns, or micrometers for or look at this one. About the same size. Again, this is just a diet. This is a single cell organism. And I argue if you came across one of those anywhere else, if you saw one of those laying on the street outside, or come across one of those on a beach, I think you could conclude that someone had formed that an intelligent mind had made that. Just because we find these in the microscopic world should not change that conclusion. I want to show you one other, but before I show it to you on the scanning electron microscope, I'll show you a video of it. This is a standard magnification. This thing coats itself with plates called coccoliths. You see, as the technician was zooming, you could see the plates that this thing is making to coat itself with. Plates made out of calcium carbonate. You see those little circular things? It's exporting one of them right now. Coats itself with these plates. I just wanted to show you what it looks like under a normal microscope. This is probably about 400 power. Standard classroom level microscopes only get up to about 400 power. So you can better appreciate what it looks like on a scanning electron microscope, which is this. I mean, there's evidence of design within the microscopic world even if you just look at it. But we know a lot more about the microscopic world now that speaks to design. And although, again, the scientific community today, by and large, argues that these things were not made, the Bible is clear on these, that these things were made. For by him all things were created, in heaven and on earth, visible and invisible. Whether thrones or dominions or rulers or authorities, all things were created through him and for him. God made all of these things. Everything that we see, the material world itself, was entirely made by him. And it's a humbling thought to realize that he made all of it for us. No other reason. The vast cosmos itself put out there just to declare to us his glory. All of the amazing beauty we see in the world around us, created just for us. Everything that you love about this world was put there just for you by a God that is infinite in his wisdom. It's a tremendous thing. But of course, again, the scientific community argues that that everything has evolved through purely natural processes. Since Darwin formed his original theory in 1859 in his book published On the Origin of Species, they've been arguing fervently for this. But it's interesting to note that at the time Darwin published his theories, almost nothing was known about the cell. I'll emphasize this a little bit. Excuse me. The first person to see a cell, again, no one was even able to see cells until we developed the technology to do so. The first person that was able to see a cell on any level did so in 1655 when a microscopic technology was developed that allowed Robert Hooke to see a thin cross-section of cork. And when he saw that thin cross-section of cork, he saw compartments in it that reminded him of the cells that he had seen in monasteries. The living quarters that were in monasteries were referred to as cells, like we refer to them in prisons as cells as well. But he said they looked like cells and coined that term cell to describe these compartments that he saw. The cell theory, the existence of the cell and how it relates to the biological world, the cell theory itself wasn't developed until the mid-1850s. Between 1837 and 1855, that organisms are made entirely of cells, and that all of the life processes are performed by cells, and all the products of a living thing are made by cells. The components of the cell theory weren't formed until about the time Darwin published Origin of Species. So the time he published his theory, it was only then just recognized that organisms are made of cells, that the life processes are performed by cells, that the products of living things are made by cells. It was hardly only known at that point in time. And they still argued for a long time, and I'll show you a good example of this, argued for a long time that cells were very simple. But of course, we know now that cells are vastly more complex than anyone of Darwin's day could have even envisioned, could have even imagined. In comparison to our marvels of modern technology like the Boeing 747, the cell has orders of magnitudes more moving parts. It is far more complex than something like a 747. It is in fact far more complex than a factory that makes 747s. It is in fact far more complex than a city. And there's really no other analogy that you can really draw to compare something with the complexity of the cell other than something as sizable as a city. It's like a city that's been miniaturized down to a microscopic scale. That's the level of complexity we're looking at there. It has factories. It has highway systems, countless machines, countless little robots running around, transporting things from one place to another. It is a complexity literally on the order of a city. But it's a city that can copy itself, that can move itself, that can make everything that's within the city and so it'd be like Seattle deciding we need another Seattle and it just making a duplicate of Seattle and one of those moving off over in the eastern Washington. An evolutionist who has turned into an advocate for intelligent design eventually described the cell this way. Michael Denton, in his book, Evolution, a Theory in Crisis, he says, to grasp the reality of life as it has been revealed by molecular biology, we must magnify a cell 1,000 million times until it is 20 kilometers in diameter and resembles a giant airship large enough to cover a great city like London or New York. What we would then see would be an object of unparalleled complexity and adaptive design. The cell is so complex, it is unparalleled in its complexity. I drew a city as an analogy to its level of complexity, but in reality, it is unparalleled in its complexity. There's nothing we can point to that is comparable to the cell on complexity or adaptive design. It is tremendously complex. It literally is like a city. The animations really don't do it justice. Just don't do it justice what's going on, because to show everything that's going on inside a cell, you couldn't see anything. It's so packed full of operations that you couldn't even really see what was going on if they showed it to you the way it really is. But just to get a kind of a look at what's happening inside, a little animation, little video animation kind of gives us a bit of a picture. So they are like cities, unparalleled in their complexity. They have a skeleton. There's a skeletal network that it can assemble and disassemble in microseconds. They don't slowly fit together like these skeletal components are assembling themselves and coming apart. It has highway systems that can assemble, self-assemble, and self-disassemble. This is a structure called a microtubule. I'll describe it again. It's got robots that carry packages down highway systems from one place to another. This is a robot called a kinesin that's transporting a big package called a vesicle down a highway system within the cell. It is literally like a city with factories and highway systems and trucks, robots of various forms, moving things from one part to another, modifying things, making things, breaking things down. There's really nothing else you can compare it to on that level of complexity other than a city. It is like a city that, again, is miniaturized down to a microscopic scale. And we're only now just really grasping the level of complexity that we're really looking at, because really we can't see it. We can't see it in action. Animations like this, what's the processes taking place in this animation were deduced. We can't see these operations happening on this scale. I use the analogy, it's like we're looking at a city from a hilltop with a telescope and trying to figure out what's going on in that city. We can see things moving around down there, and every once in a while we squish one of those cells, and we figure out these two things are stuck together, so they're doing something, and these two components are stuck together, so they're doing something, but we can't go in and look at them like this. The images that I showed you previously of the scanning electron microscope can only look at things that are dead at the highest powers, when we can really start to see details. You can't see them living, not at that level of magnification. It is a complex thing, though, a tremendously complex thing, and we're still struggling to understand all the complexity that's taking place in there. It is a wonderful thing that God has made. And multicellular organisms like us have many different kinds of specialized cells. Cells that are highly specialized. Muscle cells, for example, are specialized to shorten. They contract. They shorten, and by shortening, they move bones to help us move around. They move the stomach to help you churn food and digest food. They pump the blood in the heart. They do all the various types of movement that are necessary in the body. Nerve cells send electrical signals from one place to another, telling all parts of the body what to do. Cone cells are one of the cells in the eye that detect photons of light. The hair cells shown on the other side are one of the cells in the inner ear that are involved with both balance and hearing, or a type of cell called a hair cell. Highly different specialized forms, and in total the human body has close to 100 trillion cells of many different specialized types. And you actually make well more than 2 million cells per second. 2 million cells per second. And that is just red blood cells. I mean, you literally are making somewhere close to 2 million red blood cells per second. Just talking red blood cells. So it's a tremendous complexity that we see there. But of course, there are many today, many within the scientific community, that argue that all of what we're seeing has just come about through purely natural processes, through the scientific community is dedicated to naturalism. Now, not all scientists are atheists. I would not argue that. But the science that's being taught in our public schools, the science that's being taught to us, is definitely a science that's based on atheism. They've rejected the existence of God. They've rejected biblical history. And they're doing their best to explain how this world came to be through purely natural process. They're dedicated to naturalism. They would argue that everything came into existence through purely natural processes, and that every physical phenomena that we observed must be occurring through purely natural processes. This is what the scientific community is dedicated to. That's why they call our museums natural history museums, our science museums. We call them natural history museums. It's not just a science museum. They're teaching us a history of the world in that science museum within the boundaries of naturalism. Right? That's why we call them natural history museums. That's what we call the world that's out there nature, because they believe it's purely natural. Now, the scientists of old were referred to as naturalists. Charles Darwin himself was an unpaid naturalist who went on the HMS Beagle to do his famous voyage that went out to the Galapagos Islands and these kind of things. But unfortunately, though, a great many Christians don't understand that evolution is in direct conflict with Christianity. They don't understand how significant it is. But I would argue that evolution today is the leading cause of loss of faith, in particular amongst young people. And it is a great impediment that keeps people from coming to the faith. You can't just Can't just tell people that you believe in God and try to teach them about Jesus without them having a whole bunch of questions, because they've adopted a different worldview. People have been raised in public schools today, have been indoctrinated into a worldview that blinds them to the truth of it all. And in order to get them to change their worldview, there are a great many questions that you have to be prepared to answer. And the church is just not on top of this. The church is just not providing the kinds of education that really needs to be provided, and particularly to young people, to youth groups. It's troubling to me how infrequently I'm invited to speak at youth groups. Very troubling how rare that is. I've been invited to speak more at Slavic youth groups than I have English-speaking youth groups. True. Big Slavic youth programs, even camps, youth camps I've been brought into, but very few English-speaking youth groups. It's just, it's all about having fun and getting a lot of people in there, but very little of the kind of education that would stem a loss of faith later in life. This in apologetics is extremely important. Forgive me for laboring the point. Ernst Haeckel, shown here, was a professor at Jena University in Germany and was an evangelical promoter of Darwinism in Europe. Haeckel would eventually become infamous, notoriously infamous, for creating multiple acts of fraud that he committed in trying to prove evolution. Multiple acts of frauds committed by Ernst Haeckel to try to prove evolution. But he described a cell like this. He described what he considered to be the first cell like this. He says, it's not composed of any organs at all, but consists entirely of shapeless, simple, homogeneous matter. It's nothing more than a shapeless, mobile lump of mucus or slime consisting of albuminous combinations of carbon. He believed this was probably what the first cell was like. But where did he get this? And it's real easy to envision the origin of a first cell if that's all the first cell was, a simple little lump of mucus or slime in a little puddle of slime out there. A cell that's, if it's nothing more than a simple little lump of mucus or slime itself, it's easy for you to see how something like that developed. But had Ernst Haeckel ever seen a cell like this? No. It was pure imagination. No one has ever seen a cell like the one he described, that he called monarans, a name that was eventually applied to the bacterial kingdom. The kingdom of the bacteria was named Monera, and that's how I learned them. Since then, they've changed it to Eubacteria and Archaebacteria. The bacteria kingdom name has been changed, but it was originally named Monaran. But he never saw one of these cells. No one ever did. But that did not stop him from making more than 30 drawings of these imaginary cells, which along with 73 pages of his published speculations were published by the prestigious scientific journal in 1868. He also gave them scientific names, species names like Protoamoeba primitivia, because to him, primitive protoamoeba must have existed before the complex amoebas that we find today. So why not just go ahead and make it up until it's discovered? But the extent of detail in his drawings that you see here is really a measure of his fraud, that he could go to such lengths to make drawings of these things, talk about the reproductive processes, even though no one ever saw these things. And another example of just how fraudulent this really was of him is the fact that he knew well about diatoms. Remember, he described these monarans as simple little lumps of mucus or slime. Ernst Haeckel knew well about diatoms. He actually published on diatoms. And a contemporary of his day was actually scathing in his criticisms of Ernst Haeckel because he had some of the best microscopic technology available of his day. At Jena University, he had some of the best technology available for microscopic, detailed work. He published on diatoms. These are two of the plates published in a book that he had made in 1904. Two different plates of diatoms. And remember the diatoms that I showed you, the beautiful symmetries there. They're far from a shapeless, simple, homogeneous matter. They are far from shapeless. They have definite shapes. And he knew that. The cells that Ernst Haeckel knew of in his day were definitively not shapeless, definitively not little lumps of mucus or slime. But and he published this work, these diatoms, in a book titled Art Forms in Nature. That's the German title shown on the slide, but it's translated art forms in nature. Art forms. Now, he knew that they weren't shapeless. They were so shapely, he published in a book about art forms in nature. And isn't it interesting how a scientist committed to atheism, committed to Darwinian naturalism, could recognize art that existed within the world and not recognize that there's an artisan, an artist behind that art. You can recognize there's art in nature and still reject that there is an artist behind that art. How is that? With so much evidence. Remember, Romans 1:20 says that God put the evidence there. Romans 1:20, for since the creation of the world, God's invisible qualities, his eternal power and divine nature are clearly seen in what has been made, so that men are without excuse. His eternal power and divine nature are clearly seen in what has been made. He sees the art forms. He sees them there, but he still rejects. And if you read the rest of Romans 1:20 and 22, he says, for although they professed to be wise, they became fools and rejected God. Haeckel was also an advocate for spontaneous generation. Even though spontaneous generation, and this is from a quote in 1876, 20 years before this, Louis Pasteur had completely disproven the notion of spontaneous generation, that things could spontaneously pop into existence from meat broths or things like peanut butter or meat. They believed up until this time that things could just pop into existence, spontaneous generation. But Louis Pasteur proved this conclusively. But Ernst Haeckel was still an advocate for it, advocate for its belief, even though it had been disproven for purely religious grounds. He says, if we do not accept the hypothesis of spontaneous generation, then at this one point in history of evolution, we must have recourse to the miracle of supernatural creation. We have to believe in spontaneous generation. We have to. Because what else is left than creation? If things don't pop into existence out of nowhere, then what else is where? But it's interesting that although he called these monarans shapeless little lumps of mucus or slime, again the name monarans was eventually assigned to the bacterial kingdom, which has one of the most complex molecular machines we know of, called the flagellum. The monarans, which he described as shapeless little lumps of mucus or slime, have one of the most complex molecular machines we know of, the bacterial flagellum. Molecular machines themselves are powerful arguments for design. There's lots of examples. I'll show you a few examples of molecular machines that completely defy Darwinian explanations for their origin. And there are powerful arguments for an argument for design because they exist in a form that we describe as irreducibly complex. And Michael Behe, in his book Darwin's Black Box, kind of repopularized the notion of irreducible complexity and used in his book a mousetrap to describe the simplest machine he could think of. You really can't think of a machine any simpler than a mousetrap. But a mousetrap has multiple components that have to be there for it to function. And if you remove even one of those components, it won't work. Although it only has a few components, so any one of them is removed, it won't work. And that's the case for all molecular machines, and they are vastly more complex than a mousetrap. Michael Behe describes an irreducibly complex structure like this. He says, it's a single system which is composed of several interacting parts that contribute to the basic function, and where the removal of any one of these parts causes a system to effectively cease functioning. An irreducibly complex system, he continues, cannot be produced by gradually by slight successive modifications of a precursor system, since any precursor to an irreducibly complex system is by definition non-functional. Since natural selection requires a function to select, an irreducibly complex biological system, if there is such a thing, would have to arise as an integrated unit for natural selection to have anything to act on. It is almost universally conceded, he says, that such a sudden event would be irreconcilable with the gradualism that Darwin envisioned. These molecular machines are irreducibly complex. They simply cannot be developed through numerous successive slight modifications because it doesn't function until all the components are there. But listen to Darwin's own words. He says, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, but I can find out no such things, no such case. But of course, back in Darwin's day, they knew nothing of the cell. Nothing of the internal machinery of the cell was known at the time of Darwin. They hardly understood that living organisms were made of cells, much less that there were complex structures in cells like molecular machines. And there's lots and lots of molecular machines. I'll show you a few examples of these. Machines that do all kinds of stuff. And highway systems. Remember the animation that I showed you with that little kinesin walking one little vesicle down a highway? There are highway systems. There's not one little kinesin. Can you see the highway systems in operation on these? Whole armies of kinesins transporting their vesicles from one side of the cell to another side of the cell. Or look at this, a time-lapse video of a cell in the process of preparing itself for cell division. Countless machines in operation there, doing their jobs of getting this cell prepared for cell division, breaking down the nuclear membrane, organizing the chromosomes into distinct strands, making other proteins that are necessary for the process, then grabbing a hold of the chromosomes and pulling them apart to the different poles, and then creating a new cell wall to go between the two newly divided cells. You really get a sense of all the work that's really ongoing within a cell by seeing something like this under time lapse. But again, this is about as high as we can magnify things and watch them when they're living. Like I said, we're kind of looking at them from a distant hilltop with a telescope. What's going on down there? There's lots of stuff running around, but that's basically as much as we can do right there. Lots of work going on, lots of molecular machines. Everything that's happening there is a machine that's doing a job. And again, these cells can move themselves, can pick themselves up and move from one place to another through varieties of different mechanisms. This is an amoeba. They move through a mechanism called amoeboid movement. They can just move themselves. Every single thing within that city can just be moved. Every factory, every building, every highway can just be moved from one location to another. That's a coordinated effort we'd be thrilled to be able to accomplish. And I'll show you some of the molecular machines involved with some of this. One of them is involved with making energy for these machines. A powerful molecular machine that makes this molecule, this all-important molecule called ATP. The machine itself is called ATP Synthase. I'll show you what it looks like here in a second. But it makes this important molecule called ATP through a process called cellular respiration. The reason why we breathe in oxygen and breathe out carbon dioxide is because oxygen is needed to break down glucose. What we would think about as blood sugar. We eat plants because plants make glucose in photosynthesis. We need that glucose because it's an energy carrier. And it's broken down to make a whole bunch of these molecules called ATPs. The glucose molecule, think of it like it's a $100 bill, but your cells need quarters to do all of their work. So it breaks down this glucose molecule like this $100 bill into a whole bunch of quarters that are like ATPs, and use those ATPs for every job that needs a little bit of energy. Every muscle contraction all requires ATP. Nerve impulses all require ATP. Making molecular machines all requires ATP. Every molecular machine itself to work needs ATP. And the organelle, one of the compartments in a cell, when I was on my slide of the cell, I forgot to stop and point out the various compartments, the various components of the cell when I was on that slide. Forgive me for that. But one of the little compartments within a cell is the mitochondria. This one shown here. Sometimes called the powerhouse of the cell because it makes energy. It makes the energy carrying molecule we call ATP. And just to stress how important ATP is, if ATP synthesis is blocked by, you will die in 30 seconds. So there are poisons like cyanides. Most people have heard of cyanide. Cyanide is a very nasty poison that can kill you in 30 seconds, and it kills you in 30 seconds because it blocks ATP synthesis. And you die that quick without ATP because ATP is required for everything. Muscular contraction, nerve impulse, all this kind of stuff. And in fact, you use and remake half of your body weight in ATP every day. You use and remake half your body weight in ATP every day. Now I want to stress what's really involved with making ATP. Okay, and I don't know how well you can see this, and I can't point to this point to it up there on the screen easily because it's like behind me, but I'm going to try to get over there and point with the mouse because people online can't see them unless I point with the mouse, okay? So way up here on the top of the screen, you can't see it, but just off screen is the glucose molecule. Glucose, which again is why we eat plants because they make glucose in photosynthesis. Glucose is first broken down into 2 molecules of pyruvate. In the first part of this process, pyruvate is converted into acetyl-CoA and is converted by an enzyme. One of the all-important proteins in the body are called enzymes. Enzymes are proteins that perform chemical reactions. And extremely important, it's a molecular machine that basically performs a chemical reaction. And the reason why I'm looking at this is to point out how many different machines are necessary to do this job. And by the way, there's some quizzes I'm going to be showing you at the end, and we're going to be quizzing you on some of these names, so pay attention. Okay. So anyway, where did my thing go? Okay, so there's an enzyme name right here, pyruvate dehydrogenase, that converts pyruvate into acetyl-CoA. Then acetyl-CoA is combined with oxaloacetate, and the enzyme that catalyzes that is citrate synthase, makes citrate. Citrate is converted into isocitrate with a conatase, another enzyme. Isocitrate is converted to alpha-ketoglutarate by another enzyme right here, isocitrate dehydrogenase. This is converted into this with another enzyme. This is converted into this with another enzyme. This is converted into this with another enzyme, over and over and over. And eventually the end molecule is remade back into citrate. Okay? But lots of enzymes, lots of these molecular machines are needed to perform this part of the process of making ATP, and we still haven't made ATP yet. What this is doing, this part that we call the citric acid cycle, is actually making that NADH right there, and it's making an NADH right here, another NADH right here, because those molecules are needed to power this. Those and the hydrogen ions that they make are used to power this. This is the ATP synthase, which was just discovered in like the 1990s. The guy that discovered this won the Nobel Prize in 1997 for discovering the ATP synthase. And there's like, I don't remember how many ATP synthases there are in a mitochondria. I need to look that up. But it works through proton motive force. Protons flow through this thing, and as protons, or the hydrogen flows through this machine, it spins, the head of that thing spins at 200 times per second, and every time it spins, it makes 3 ATP molecules, okay? Shown here in a little different kind of a diagram, showing how many individual proteins are used to make this one machine. This has 500 individual protein subunits that have to be assembled together to make this one machine that makes ATP. So you have to have the ATP synthase. You have to have all of the enzymes that are required for the citric acid cycle and what we call glycolysis, the first part of that. And to make matters worse, the evolutionists have to explain this is an all-important process performed by every organism on Earth, essentially. They all do this. And evolutionists have to explain the origin of a complex series of chemical reactions like this, and something as complex as this proton motive force little machine we discovered in the mitochondria. And this entire process still requires ATP. You have to have ATP. To make ATP, you have to have the enzymes to make ATP. You have ATP synthase to make ATP. And evolutionists are challenged with the job of trying to explain how all of this came about through purely natural process. Let me show you another pretty cool molecular machine. It's called the cilia. This is a ciliate. There are a number of organisms. This is a single-cell organism that has cilia that it uses to swim around. It also uses to eat. Watch this right quick. You'll see for a second that it eats something using cilia right here. Watch out. You see it gulp down a little piece of algae right there. They're covered with cilia. You can see the cilia moving on the outside. The cilia move in a wave-like manner, kind of like people wave in a stadium. They move in an orchestrated wave-like manner to swim through waters, aqueous solutions, and they also cover their mouth to help channel stuff into their mouth when they're ready to eat. So it's called cilia. It's a complex molecular machine that they're covered with these things, and there's lots of ciliates, different kinds of ciliate. This is a ciliate that's inside of the gut of a termite. So the reason why termites can eat your house, because they got a bunch of these things in their guts helping them digest your 2x4s. But look at this, it even has a head. This is a single-cell organism, but you see it has a head, even though it's a single-cell organism, but covered in cilia. That wave-like hairs, those are the cilia. You actually have cilia too. So your lungs, the air passage in your lungs are covered with cilia also. You have cells in your air passages that are making mucus, what tends to come out as, mucus, right? And so they make mucus, and then they have cilia that constantly move that mucus up out of your lungs, out of your air passages, to clean your lungs. Your lungs are self-cleaning. So when you breathe in air, all of those little fine particulates that are in the air will get stuck to the mucus, and the mucus is moved up out of your air passageways, where you can blow it out your nose, but more frequently than not, you swallow it. It brought up out of your bronchioles, it gets to your esophagus, and you swallow all that. And actually intending to gross you out with this, I was getting ready to say I wasn't intending to, but actually I am intending to gross you out with this. You know that most of the dust in your house, most of the little particulates floating around the air, most of the dust that settles out in your house are dead skin cells. And you're basically filter feeding the air of dead skin cells and eating dead skin cells of everyone that lives in your house. So, in one way, cannibals. Cannibals eating the dead skin cells of all the other people in your house. It's true. It's true. It's very, very true. Okay, but the cilia is a complex molecular machine. Let me point out some of the internal structures, the components of cilia. This is a cross-section of a cilia. So cilia is a long hair-like structure, okay? And if you took that hair and cut it off, this is a cross-section of a cilia. And you see the little circles in there. There's 9 pairs of 2 circles. Those circles are microtubules. In the video that I showed you earlier, that the walking robot was walking down, that was that tubular-shaped thing that was assembling and disassembling, that thing was self-assembling, that's a microtubule. Microtubules are used as highway systems in the cells, that things like kinesins can walk down. They are what reaches out and grabs a hold of the chromosomes during cell division and pulls the chromosomes apart. And in this case, they're a structural component of the cilia. Okay, those circular things are those microtubules. And it is a complex molecular machine, the cilia is. Look at this diagram of a cilia. Kind of reminds you of an engineering diagram, doesn't it? That's because any accurate diagram of a molecular machine definitively looks like an engineering diagram for a very obvious reason. They were engineered. It's an engineering diagram because they are engineered, and if they're engineered, there's an engineer behind them. Just like there's an artist behind the art, there's an engineer behind the engineering. And they most definitively, any of your detailed diagrams of molecular machines look like engineering diagrams for that reason. Here's a little bit different diagram of cilia. Let me point out a few of the functional parts of this. That again is the cross section that shows the 9 pairs of microtubules and then another pair of microtubules right in the center. Let me point out a couple of the motor proteins that are needed for this. There's one, so the microtubules have little motor proteins attached to them called dienes. What they do is one of these motor proteins basically reaches out, grabs a hold of the neighboring microtubule and pulls on it, a process requiring ATP. Then there's another important protein part of this that's called a nexin. That's right there. And this is used to attach the microtubules together. So you got a motor that reaches out and grabs hold of a microtubule and pulls on it. The other attaches them together. And this is very important because as this diagram tries to illustrate there, these are those pairs of microtubules standing next to one another. And if the diene pushes down or pulls on the neighboring microtubule, they would slide one next to each other. But what you want is the structure to bend, because that's how cilia work. It's a wave, produces a wave-like manner in order to push water. So what the diene protein also needs the nexin protein locking the microtubules together, converting the sliding motion into a bending motion. But multiple components are necessary here. Multiple components are necessary for the cilia to work. Michael Behe stresses this, again in his book, Darwin's Black Box. He says, what components are needed for a cilium to work? Ciliary motion requires microtubules. Otherwise, there would be no strands to slide in the first place. Additionally, it requires motor, or else the microtubule of the cilium would lie stiff and motionless. Furthermore, it requires the linkers, the nexins, to tug on neighboring strands, converting the sliding motion into bending motion and preventing the structure from falling apart. All of these are required to perform one function, ciliary motion. But again, Darwinism requires that everything is developed through numerous, successive, slight modifications over countless generations. You have to make something, you have to make one part of the machine, and it has to have a function. And you keep making it just like the mousetrap. So think about the mousetrap analogy again. For that mousetrap to be built by Darwinian mechanisms, you have to have one piece of the mousetrap made, only one piece, and it has to have a function. It's a mousetrap, but it's not a good, it's just kind of not a really good mousetrap. And then you add one more part of it, and it's a little more functional. It's still a mousetrap, but a little more functional than a mousetrap. You add one more part, it's still a little more functional, but it's a little better mousetrap. And but it has to have a function at each and every stage in its development, and it has to be better as each and every component is added to it. This is how Darwinian mechanisms, Darwinian natural selection process must work. But the vast majority of these molecular machines, I only say vast majority, I'd say all these molecular machines have multiple components, and the removal of any one of those components would cause the machine to stop functioning. Certainly the case here with the cilia. You can't get a cilia through numerous successive slight modifications. You have to have everything there at the exact same time for it to work. Another similar mechanism in the cilia is called a flagellum. It's a similar strand-like motor that is used by the cell, but instead of moving in a bending fashion, a wave-like fashion, it spins. This is a flagellate. So cells that have flagellums are called flagellates. This one's extending its flagellum, and then it'll spin its flagellum to push itself through water. Others pull themselves through water with the flagellum. This is another flagellate spinning its flagellum and pulling itself through water with its flagellum. Okay, similar to the cilia, except it spins rather than bends. But bacteria also have these. And remember, it was the bacteria that were given the name monarans, because they were believed to be the simplest organism on earth, but it come to find out, bacteria have one of the most complex molecular machines we know of. The bacterial flagellum is a tremendously complex machine possessed by these little bacteria. These are bacteria swimming around using their flagellum, and they swim incredibly fast. They swim actually at 60 cell lengths per second. And they swim at 60 cell lengths per second, but I went ahead and did a comparison of this just to figure out how fast they're really moving, because it looked like they were moving really fast. So how fast is 60 cell lengths per second compared to another animal that's like really fast? Cheetahs move at 25 body lengths per second. So cheetahs move 25 body lengths per second. That's still crazy fast. Think about that. That a cheetah can 25 body lengths per second, but the bacteria through its flagellum is moving at 60 cell lengths per second. By comparison, that's like 164 miles per hour. If it was the size of a cheetah, that is. It'd be moving at 164 miles per hour. It's still moving pretty fast with this flagellum, but it is a molecular machine, a complex molecular machine. David Rosier describes it as a machine design resembling a machine designed by humans. If it resembles a machine designed by humans, why aren't we starting to think about whether or not it was designed by someone at least as intelligent as humans? Why? This is how science is supposed to work, you know? You're supposed to use your experience to inform your observations and help you reach your conclusion. But today, we're not really thinking about our experiences. Our experience and observations in science count for almost nothing when it comes down to evolution. Their worldview dominates. Evolution is regarded as fact, even though how it happened is still highly hypothetical. We call it the theory of evolution, whether it's through Darwinian natural selection, whether genetic information is a result of random mutations, whether these theories are accurate or not is still up in the air. But the theories are still being worked out, but that doesn't stop them from calling it a fact. But that's the scientific method flipped on its head, because you're supposed to start with a hypothesis, an educated guess, and then you test that hypothesis with experimentation, and eventually you may be able to elevate that hypothesis to a theory. After some experimentation, you elevate your hypothesis to a theory. Then you do more experiments, and you do experiments specifically designed to falsify your theory. It's what they call Popperian falsification. You actually design experiments to try to falsify your theory. And if your theory cannot be falsified under any conceivable circumstances, if it's shown itself to be true under every possible conceivable scenario, then you might be able to regard it as a fact. But they've taken evolution and just flipped the whole scientific method on its head. We're going to start with the assumption that it's a fact, Assumption is too soft. It is a fact. Evolution is a fact as far as they're concerned. Now let's work out the theories to see how it happened. But I digress. Back to my point, to my subject here. The bacterial flagellum is a machine that looks like it was designed by humans. It's the world's smallest rotary propulsion system. That's what it is. It has all the parts that we would place in a motor. It is constructed of proteins, that we can't do that, but each of these proteins has different functions, such as those that serve as there's a protein that serves as the rotary motor itself, working through a proton motive force similar to the ATP synthase. You have other proteins that serve as bushings. There's one that serves as a drive shaft. There's proteins that serve as a rotation switch regulator. It has a universal joint. It has a helical propeller, and it has a rotary promoter that allows it to self-assemble. It's a motor that self-assembles. It's a motor that self-assembles. It has all the parts that we would place in a motor, plus it can self-assemble. At least the filament itself self-assembles. This animation was made by a company in Japan that was looking at this in a hope that they could copy it. Works through proton motive force. You can see the protons flowing through this in this animation. But it's a nanomachine that is coded by information from approximately 50 different genes, and is composed of somewhere between 25 to 40 different structural proteins, depending on the species, each of them with multiple copies ranging from just a few to tens of thousands. The long tail itself there is made up of tens of thousands of proteins that are assembled together in that long chain. In every way, it truly resembles a machine designed by humans, like David Rosier, the flagellar expert, said. If it looks like a machine, it is a machine. And if it looks like it's a machine that was designed, there must be a designer behind it. And that should lead us to the conclusion that there is a designer behind these things. But evolutionists assert that the bacterial flagellum was developed through a process called co-option. Let me check my time and just make sure I'm not running on a bit here. I think I'm okay. They say it was developed through a process called a co-option. And this is one of the classic evolutionary explanations. They argue that it was not developed through numerous successive slight modifications necessarily, because all it did was take pre-existing parts and repurpose them. And this is argued for the bacterial flagellum. There are a few parts, just like we tend to use parts that are being used for other things. If you look at the motor, if you look at your engine itself, you can find a few bolts or whatever, a few O-rings and things that are used by other motors, that are used on completely different kinds of constructions. So we do the same thing. And within cells, there are some parts that are shared. It's just a basic structural protein that's used on more than one kind of device. God has done this as well. And so on the flagellum, there are a few proteins that are found elsewhere in the cell. And one of the places where we find a couple of flagellar proteins is on this little device right here, which is called the type 3 secretory system, which is itself like a harpoon, if you will, or a spear that bacteria use to inject poisons into cells that they're wanting to kind of speed along into the decomposition process. Like bacteria are decomposers, right? But there's a few bacteria that get a little aggressive about it and decide, you're not really dead yet, but let's see if we can't go speed that along. And this is the bacteria that caused the bubonic plague. Use the type 3 secretory system to inject poisons in and cause a huge plague at that point in time. But the type 3 secretory system does have a few proteins shared with the flagellum, but a full 75% of the proteins in the flagellum are completely unique to the flagellum. 30. It has 40 structural proteins total, the bacterial flagellum does, and 30 of these, full 75% of the proteins used by the bacterial flagellum are completely unique to the flagellum, found nowhere else in the cell. Yeah, a few are shared. A couple are found there in the type 3 secretory system, but 75%, 30 of the structural proteins used by the flagellum are completely unique to the flagellum. The co-option argument just falls flat. One of the most powerful arguments for intelligent design has to be the discovery that information exists within cells. And this is such a powerful argument because in all human experience, everything we know about information tells us that information comes from only one source, and that's an intelligent mind. In all human experience, information comes from only one source, an intelligent mind. So when we discovered that there was information in cells, it should have led to the conclusion that the cell was also the product of an intelligent mind. Because again, this is how the scientific method is supposed to work. But instead, following that discovery, evolutionists have made resolute that information must also come about through purely natural processes. So one of the biggest challenges to evolution today is the existence of genetic information, the origin of genetic information, the origin of life itself. We cannot explain how a cell came into existence all by itself. They can't even explain how a DNA strand could develop all on its own, how a single protein could develop all on its own. We're light years away from explaining how something as complex as a cell could have developed all on its own through purely natural processes. But the existence of information itself is arguably one of the most powerful arguments for intelligent design because, again, in all human experience, information comes only from an intelligent mind. And yet we found it there within cells. And where we discovered it was in the nucleus of cells. Again, this is that diagram of the cell I showed you previously, although I forgot to point out the structures there, like the mitochondria, one of the compartments there. You have other compartments like the chloroplast and the mitochondria and Golgi body and endoplasm reticulum, all this kind of stuff. And right in the very center is a large structure called the nucleus. The nucleus is the storehouse of your genetic information. The molecule that we call DNA, deoxyribonucleic acid, which looks like a ladder that's been twisted, so it's a shape that they call a double helix. And what DNA is a complex coded instruction. This code is made up of 4 different components, 4 what we call nucleotides. It's a long strand. Each rung of the ladder, if you will, each step along that ladder is made up of a different molecule, one of 4 different molecules that are represented by the letters A, T, C, and G. What you see next to that DNA ladder is actually a DNA sequencing gel. This is one of the tools that is used to determine the sequence of these A, T, C's, and G's and on a DNA strand. Sequencing information that often looks like this. So it's a huge sequence of these letters, A, T, C's, and G's, that is read by the cell, and it is instruction that the cells follow. But just to impress upon you how much information you're looking at there, if every letter A, T, C, and G represents one of these 4 nucleotides on this DNA strand, if every letter there is equivalent to a letter in our alphabet, then there is about as much information in one cell as there is in 1,000 books. If each A, T, C, and G in that DNA strand is equivalent to one of the letters in our alphabets, then you're in one human cell, there's about as much information as there are in 1,000 books. Or to put this another way, a 2-millimeter pile of DNA, or a pile about the size of a pinhead, would have about as much information as 500 stacks of books reaching the moon, or a single stack of books reaching the sun. It's an incredible amount of information that's been found there by way of this DNA strand. And evolutionists are faced with an enormous challenge of explaining how that information came about. And one of the most ludicrous suggestions put forth by evolutionists is that this genetic information came about by just random changes. That you had a very simple organism in the beginning, and by randomly changing the genetic information of that organism, you eventually got more information. It's a ludicrous explanation, a ludicrous suggestion. It's an affront to reason, it's an affront to logic, and it's an affront to common sense to suggest that you can randomly change information and create new information, create more information. Like taking a book off of your shelf and randomly changing letters on that book could eventually make more information. You can get more books that way. Or by randomly changing the blueprint or the assembly instruction on my big old tube TV that I still got at home. I still got one of these big old tube TVs. Thing's so big and heavy I can hardly lift it at this point. But, I would love if I could make random changes on the blueprint of that thing and get me a big flat screen TV. But the fact is, you can't. It's a ridiculous assumption to claim that you can randomly change information and create more information. That by randomly changing the genetic code, you went from a simple organism, and you can't even call bacteria simple, right? You saw the bacterial flagellum, that you can't go from an organism like bacteria to something like us. By just randomly changing information, you can't create more information and go from, and I don't know if I can reuse the word less to more complex, because the bacteria itself is vastly complex. It's a single cell that can do all the things a multicellular organism can basically do. It's hard to even use the word simple to complex anymore when you're talking about the biological world. But that's the explanation that's given. All right, let me explain a little bit more about how DNA is used, what its function actually is within cells. Now, DNA exists as a long strand we call chromosomes. The DNA helix that you saw there, if that exists as a really, really long strand. Humans have 46 chromosomes that range in size from 1.7 to 8.5 centimeters in length. If you took all of the DNA in one of your cells and attached them end to end, it would be a strand about the size of about 6 foot in length. Okay, but one of those strands, when it's coiled up for cell division, looks like the structure you see there. It's typically how we see chromosomes. But one of these long strands of DNA is what we call a chromosome, and a chromosome is this long strand. If unraveled, you see that DNA, that helical structure, and it contains reading units called genes. Humans have, again, 46 chromosomes. It's estimated that we have about 30,000 genes. And the gene is a specific portion of this coded instruction, an instruction that ultimately tells cells how to make proteins. Now, that's the general definition of that. It tells cells how to make proteins. It also tells them what to do with the proteins, where to send them, how to package them, and all these kind of things. But proteins are the machinery and the material that make life possible. So all of the molecular machines that I showed you, the bacterial flagellum and the ATP synthase, every machine, all of the enzymes that were involved with cellular respiration, all the work that's done in cells is done by proteins. Your muscles contract because your muscles are made of proteins, and these proteins are loaded with molecular machines that push these filaments so they slide past each other, just like the microtubules of the cilium slide past one another. So DNA is very, very important because it provides instruction that tells cells how to make proteins. And it's the proteins that are responsible for doing all the work that's necessary to be a living thing. And it is a coded information. Even Bill Gates said it's by far the most sophisticated program around. Everyone recognizes that it's a programming code that we're looking at there. Let me show you a little bit about how it works as I move towards a close here. Just want to give you a little more information on it. What the DNA is made of, I already told you that it's made of 4 different nucleotides that are represented by the letters A, T, C, and G. These nucleotides are read by the cell 3 at a time. And 3 nucleotides at a time make up a little mini coding unit called a codon. It's a code. They're called codons. And the cell will read these, the DNA strand, 3 nucleotides at a time, and it tells the cell the order of amino acids to put in the protein chain that it's making. But it has other instructions there. It tells it what to do with the protein afterwards, where to take it, how to package it, and all this kind of stuff. But for example, if UGC is found in the DNA code, that tells it to put cysteine in the protein next. If CUG is there, it tells it to put the amino acid leucine in the protein next. AGU is the code for cyranine. GCA is the code for alanine. So it's a code that, amongst other things, tells it how to make a protein. The order of amino acids assembled together to make important proteins like ATP synthase. Remember, ATP synthase was made up of 500 individual protein subunits. So it takes 500 genes to make 500 individual protein subunits to make up something like ATP synthase, or to make up something like this is the hemoglobin. Hemoglobin is the protein in red blood cells that binds and carries oxygen, and there are 270 million hemoglobin in every one red blood cell. Francis Crick was one of the co-discoverers of the DNA helix. James Watson and Francis Crick won the Nobel Prize for discovering the shape of the DNA helix. Francis Crick said, "Biologists must constantly keep in mind that what they see was not designed, but rather evolved. This is the world that we live in, a world where scientists cannot see the evidence of design that's all around them. There's enormous evidence for intelligent design. If we just look at cells microscopically, we can see evidence of design. All the various molecular machines that we see, like the ATP synthase or the cilia, or flagellum, the existence of biological information cries out for a creator, cries out for a designer. And yet the scientists of today refuse to see it. Again, Paul spoke to this in Romans 1:20. He says, "For since the creation of the world, his invisible attributes, his eternal power and divine nature have been clearly seen, being understood from what has been made, so that they are without excuse. For even though they knew God, they did not honor him as God or give thanks, but they became futile in their speculations, and their foolish hearts were darkened. Professing to be wise, they became fools." That's the world we live in. There's enormous evidence for design, but scientists today have become futile in their speculations. Professing to be wise, they became fools. Let me close out in a word of prayer. Father, thank you so much, Father, for the wonderful creation that you have made. Father, again, we ask for wisdom. Father, we want to serve you. We want to be witnesses for you. But sometimes we find ourselves intimidated by people that know more than us. Father, we need wisdom. Father, and I ask you to empower us to speak when the opportunity presents itself, to speak to the people that we know who need to hear the testimony that we have, the testimony about your creation, the testimony that you, Father God, are the creator of the universe, that you have made this wonderful world, that you have made it for us, and that you have made us for a purpose, and that you have sent your Son to die for us because of our many sins. Father, embolden us to testify when opportunity presents itself. Father, give us wisdom, Father. Walk with us this week and help us to find opportunities to testify, we pray. All things we ask in the name of your Son, Yeshua. Amen. Go with God.